Greetings, and welcome to the lecture on plant molecular biology. Today, we will explore the topic of somaclonal variation in plant tissue culture. Somaclonal variation refers to the genetic variation observed in plants produced through plant tissue culture, or regenerated from cultured somatic cells. Plant tissue cultures, derived from a single explant, can exhibit variation after repeated subculturing. Several terms are used to describe plants regenerated from different tissue cultures, such as caliclones for those regenerated from callus, and protoclones for those regenerated from protoplasts. Similarly, gametoclones refer to plants regenerated from haploid cells in culture. To distinguish the types of variation arising from specific cell or tissue cultures, the variations are named according to the culture type. Protoclonal variation occurs in protoplasts, gametoclonal variation in anthers and microspores, caliclonal variation in callus, mericlonal variation in the apical meristem, and somaclonal variation in leaves, stems, roots, or other somatic tissues. In a broader sense, somaclonal variation also includes changes observed in cell cultures that have been used to select cell lines tolerant to extreme environmental stresses, resistant to diseases, or for higher productivity of industrial phytochemicals. Its occurrence can be both a boon and a bane. On one hand, somaclonal variation can produce desirable genotypes, resulting in novel cell lines, or plants, with agronomic and commercial advantages. On the other hand, when the objective is clonal multiplication of valuable true-to-type elite clones, this variation can result in unwanted off-types that reduce the commercial value of the propagules. Similarly, genetically engineered plants are carefully screened to avoid unwanted and unintended somaclonal variation, ensuring that the commercially released clones exhibit only the beneficial effects of the transgene. Somaclonal variations can be genotypic or phenotypic, with phenotypic variations being either genetic or epigenetic in origin. Typical genetic alterations include changes in chromosome numbers, such as polyploidy and aneuploidy, changes in chromosome structure caused by translocations, deletions, insertions, and duplications, as well as alterations in DNA sequences resulting from base mutations. Typical epigenetic-related events include gene amplification and gene methylation. If no visible morphogenic changes are apparent, other plant screening procedures must be applied to detect somaclonal variation in plant tissue culture. One of the more frequently encountered types of somaclonal variation results from changes in chromosome number, namely aneuploidy, polyploidy, or mixoploidy. Aneuploidy refers to a numerical change in part of the chromosome set. It is characterized by the presence of an abnormal number of chromosomes in a cell, where there is a difference of one or more chromosomes, but not a complete set. In contrast, polyploidy refers to a numerical change in the entire set of chromosomes. This condition occurs when a normally diploid cell or organism acquires one or more additional sets of chromosomes, resulting in 3 or more times the haploid chromosome number. Mixoploidy, on the other hand, refers to an organism that has an unequal number of chromosome sets in adjacent cells or tissues. For example, this occurs when two cell lines, one diploid and one polyploid, coexist within the same organism. Changes in ploidy originate from abnormalities that occur during mitosis. For example, these abnormalities can include extrachromosomal duplication during interphase, spindle fusion, or a lack of spindle formation, and issues with cytoplasmic division. As plant cells grow and age, the frequency of changes in ploidy increases. Therefore, changes in ploidy observed in cultures and regenerated plants may originate from the source of the tissue explants used. Another cause of variability due to changes in ploidy is the in vitro culture regime itself. The longer the cells remain in culture, the greater their chromosomal instability becomes. In addition, the composition of the growth medium can trigger changes in ploidy. For example, both kinetin and 2,4-D are implicated in ploidy changes, and cultures grown under nutrient limitations can develop abnormalities. Selecting a suitable explant and an appropriate culture medium can therefore enhance the chromosomal stability of the culture. However, high variations in ploidy in cultures do not always lead to high frequencies of somaclonal variation in regenerated plants. This is because, in mixed cultures, diploid cells appear to be better suited than aneuploid or polyploid cells for regeneration, as they are more likely to form meristems. Structural changes in nuclear DNA appear to be a major cause of somaclonal variation. These changes can modify large regions of a chromosome, and may therefore affect one or several genes at a time. These modifications encompass various gross structural rearrangements, such as deletions, that result in the loss of genes. Inversions, that involve alterations in gene order. Duplications, that refer to the duplication of genes. And translocations, that involve segments of chromosomes moving to new locations. The activation of transposable elements can also cause somaclonal variation. Transposable elements, or transposons, are mobile segments of DNA that can insert into coding regions and cause gene disruption. Tissue culture-induced mutations associated with transposable element activities have been reported in many plant studies. These mutations are related to stress conditions during tissue culture, such as wounding, exposure to hormones or specific compounds in the growth media, and genomic shocks resulting from cytological changes. Transposable elements and retrotransposons can function as insertional mutagens in plant genomes. Widespread activation of these elements can result in numerous chromosomal rearrangements, leading to the misregulation of genes, aneuploidy, and new transposon insertions. In addition to larger modifications of the nuclear DNA sequence, changes at the level of a single DNA nucleotide that occur in a coding region can also lead to somaclonal variation. For example, point mutations that result from a change of base in a single nucleotide, or the altered methylation of a base, can lead to gene inactivation. Epigenetic changes that cause somaclonal variation can be temporary or transient, and are often reversible over time. However, they can sometimes persist throughout the life of the regenerated plant. Epigenetic changes may be caused by DNA methylation, DNA amplification, or the activation of transposable elements or transposons. DNA methylation is important for both transcription and translation, because it alters the substrate affinities of enzymes that are active in these processes. Tissue culture media can change the level of DNA methylation, and may thus be one of the important causes of somaclonal variation. Over the years, considerable research has been conducted to identify the causes of variation in culture. Apparently, every possible factor that could result in a genetic change has been accounted for as a cause of somaclonal variation. This variation may also occur due to changes in organelles, DNA, enzymes, protein profiles, and the cytoplasmic genome. Factors that contribute to somaclonal variation are categorized into physiological, genetic, and biochemical categories. Variations induced by physiological factors were identified quite early. Such variations include those induced by habituation to plant growth regulators in culture and specific culture conditions, and they are often epigenetic. These variations may not be inherited in a Mendelian fashion. Prolonged exposure of explanted tissue to powerful auxins, such as phenoxyacetic acid or 2,4-D, often results in variation among the regenerants. Tissue culture regenerants exhibit certain variations that result from alterations at the chromosomal level. Although the explanted tissue may be phenotypically similar, plants often contain tissue comprised of diverse cell types. In other words, there is cytological variation among the cell types within the explanted tissue, and this pre-existing condition often leads to the regeneration of plants that are dissimilar. These species are referred to as polysomatic species. Chromosomal rearrangements, such as deletions, duplications, and somatic recombination, are the chief sources of genetic variation exhibited by somaclones. In tissue culture, several biochemical variations have been identified in various crop plants. Some of these variants show Mendelian inheritance, but many may be epigenetic and could be lost in the regenerated plant. Biochemical variations include alterations in carbon metabolism that lead to a lack of photosynthetic ability, changes in starch biosynthesis, and disruptions in the carotenoid pathway, as well as variations in nitrogen metabolism and antibiotic resistance. While genomic DNA typically exhibits normal methylation patterns, prolonged exposure of plant tissue to in vitro culture may result in the alteration of these normal methylation patterns. Studies concerning different aspects of somaclonal variation are important for several reasons. First, it is regarded as a novel source of genetic variation, and its successful utilization depends on systematic evaluation and judicious use in breeding programs, which requires appropriate experimentation. Second, somaclonal variation is of interest as a basic genetic process, because it contradicts the concept of clonal uniformity. It is believed that the cell undergoes stress, which leads to the genome, known for its plasticity, restructuring itself to modulate gene expression as required by in vitro conditions. Third, somaclonal variation is undesirable when the objective is the micropropagation of elite genotypes or genetic transformation, which partially involves tissue culture. Minimizing variation is necessary, which can be achieved through manipulation of media components, explant sources, culture conditions, and so on. Mutagenic agents, both chemical and physical, such as ultraviolet radiation, ethyl methanysulfonate, nitrous guanidine, and sodium azide, are used to produce various heritable forms of somaclonal variation. UV radiation induces dimer formation between adjacent thymine residues in DNA. This process produces lesions that can cause frameshift mutations and base pair substitutions during DNA replication. EMS and nitrosaguanidine are alkylating agents that cross-link and sever DNA molecules, often resulting in significant DNA alterations. In contrast, sodium azide induces single base pair substitutions, or deletions, resulting primarily in small point mutations. There are several different approaches to detect and isolate somaclonal variants from cultured plant cell populations. Morphologically distinct cells, such as non-photosynthetic or non-green cells, as well as cells that accumulate anthocyanins or other plant pigments, can be detected visually. To isolate herbicide and antibiotic-resistant variants, plant cells are grown on media supplemented with herbicides or antibiotics. This method can eliminate wild-type cells and allow herbicide or antibiotic-resistant variants to survive. The direct selection technique can also be used to isolate temperature-tolerant variants, as cells that survive extended incubation periods at abnormally high or low temperatures are considered temperature-tolerant. Somaclonal variants that cannot be detected visually or selected directly are isolated by indirect means, such as auxotrophic plant cells, which are unable to synthesize a particular organic compound required for their growth. When grown in the absence of the necessary nutrient supplement, these cells become very weak. In this weakened state, they assimilate exogenously supplied cytotoxic compounds, such as arsenate, bromodioxyuridine, or fluorodeoxyuridine, at a lower rate than wild-type cells. Thus, treating a cell culture under restrictive growth conditions with one of these substances will tend to favor the short-term survival of the weaker auxotrophic cells, while this restrictive condition will not affect the normal wild-type cells. In conclusion, several strategies have been employed to determine the genetic fidelity of in vitro produced progenies, considering that the commercial viability of micropropagation technology relies on maintaining genetic fidelity in the regenerated plants. While tissue culture-induced variations pose a significant threat to the genomic integrity of regenerated plants, they also provide plant breeders with tools for improvement, especially for crops with a narrow genetic base. With that, the presentation will come to an end. It is hoped that the lecture was informative. Thank you very much for attending and participating in this lecture. Thank you.